So this is an enlargement of what we were just looking at. So this outside structure is called the Bowman's capsule. The inside capillary bed here is called the glomerulus. And this entire round thing then is a renal corpuscle. And so we filter blood and produce a fluid that's going to collect in this space called capsular space. And then we're going to process that fluid to make urine as we go along. So to create a filter, we have to have a layering system. So we have blood flow into this capillary bed, and we have blood flow out of the capillary bed. So notice that this is bright red and this is more of a purple, which indicates that oxygen was used in this area here. So we're going to use words that we used in the nervous system to describe a relationship to the glomerular apparatus. So remember, in the nervous system, if it was going to the central nervous system, it was afferent. If it was going away from the central nervous system, it was efferent. So since blood is going to the glomerulus in this blood vessel, it's the afferent arteriole. Since this blood vessel is draining this capillary bed, it's the efferent arteriole. And then the reason why it's not red is because we used oxygen in this process. So if you recall way back when we did epithelium, then we said the entire cardiovascular system was lined with simple squamous. In the heart, we call it the endocardium. In a blood vessel, we call it the endothelium. So this red layer represents the simple squamous cells that make up the endothelium of the capillary bed. The squamous cells actually have little pores between them, which we call fenestrations, that you can't really see on the model. And the fenestrations allow anything but blood cells to pass through it. So if you'll recall, all epithelium sits on a basement membrane. So in this model, purple represents a basement membrane. So the purple here is the basement membrane of the glomerulus, this capillary bed. And then the purple here is the basement membrane that this simple squamous of Bowman's capsule is sitting on. So in this instance, the second layer of my filter is the basement membrane, and it limits large proteins from passing through. So with this layer of the filter, we prevent the loss of blood cells, and with this layer of the filter, we prevent the loss of large proteins. Now, sitting on top of the basement membrane to the outside of this glomerulus are cool little cells represented here that are called podocytes. So site for cell, poda for foot. So these podocytes have these little cell extensions on them. And then between the little white extensions, there's a little opening we call the filtration slit. And then there's a little membrane that covers this opening called the slit membrane. Now, the slit membrane prohibits the loss of medium proteins, so that when fluid is passing through this filter, the endothelium prevents the loss of blood cells. The basement membrane prevents the loss of large proteins, and the slit membrane prevents the loss of medium proteins. And then anything else in your blood, sugar and all this other stuff, can actually exit and become part of the filtrate that's out here. So one of the reasons why we have to have this convoluted tube, the proximal convoluted tubule, is that sugar, amino acids, and other things that we don't want to get rid of actually need to be picked up. So a classic example would be sugar. In normal physiology, sugar is not common in urine unless you were really hyperglycemic when you gave your urine sample because you'd had a tremendous load of sugar by eating candy or something right before. And as long as insulin is present, these cells pick up sugar and don't let it enter the urine. So in diabetics that don't produce insulin, these cells don't recognize sugar, and you end up with sugar in the urine, which is why diabetics who don't have controlled insulin yet have urine that has a real sweet kind of tutti frutti smell to it. And years ago, the way we would know if somebody had diabetes is somebody would stick their finger in a urine sample and taste it with the tip of their tongue. If it smelled really sweet, we know the person had diabetes. So the other thing we want to look at is that relationship that we're talking about here, where the distal convoluted tubule passes over this blood vessel. So this blood vessel would be carrying blood to the glomerulus, so our afferent arteriole. This blood vessel coming away from it here is the efferent arteriole. So here is our distal convoluted tubule. And then here is our afferent arteriole here. And it could be the efferent arteriole, it could be both. There's variations. So where the distal convoluted tubule sits against the arteriole, this whole structure is called a juxtaglomerular apparatus. Now, if we look at the lining of the distal convoluted tubule, it is cuboid epithelium, simple cuboid epithelium. But notice on this side, the cells are longer, so they look more like columnar cells. So this special row of cells here is called a macula densa. And the macula densa actually responds to changes in the filtrate, or what will become urine that's in this tube. And if we get a change in volume, if we get a change in ion concentration, then these cells communicate with cells that are in the wall of the arteriole. So the macula densa cells communicate with these cells in the wall of the arteriole that are called juxtaglomerular cells. And then juxtaglomerular cells are going to create a enzyme that's going to be dumped into the blood, which is called renin. Now, in the last unit, you did renin as an enzyme that made cheese. This renin is not the same enzyme. So the enzyme that makes cheese is Renin. This enzyme is Renin, so one N. And what it's going to do is create a change in the blood with several molecules that are eventually going to help control the activity of the kidney itself. And that'll be covered in lecture.